When people hear the word CRISPR, most immediately think of one thing: genetically engineered babies. Designer DNA. A future where parents pick their child's eye color, height, intelligence, even personality before birth. It's a concept that both fascinates and terrifies us. After all, the idea of editing human embryos hits something deep in our moral instincts. Are we playing God? Are we rewriting evolution? But the truth is, CRISPR isn't just, or even primarily, about editing babies. That's the part that grabs headlines, yes, but it's only the tip of the iceberg. What CRISPR is really about is a revolution in biology and medicine that touches everything from cancer to agriculture, from genetic diseases to endangered species. And unlike the baby editing hype, these applications are already saving lives today. To understand why CRISPR is such a big deal, we need to understand what it actually is. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a natural system that bacteria have used for millions of years. It's their defense mechanism against viruses. When a virus attacks, bacteria can snip out a piece of the invader's DNA and store it, like a mugshot in a molecular database. The next time that virus shows up, the bacteria recognize it and send out an enzyme, often Cas9, to find and destroy it. Scientists realize that this mechanism, which evolved in single-celled organisms, could be repurposed. If you can guide that enzyme to any DNA sequence you want, then in theory you could edit any gene in any living organism. And that's exactly what CRISPR-Cas9 allows us to do. Imagine it like a pair of molecular scissors guided by a GPS. The guide RNA leads Cas9 to the exact spot in the genome that needs editing, and Cas9 makes a cut. After that, the cell's natural repair systems take over. Sometimes scientists let the cell patch it up imperfectly, which is useful when you want to knock a gene out. Other times they provide a template so the cell can rewrite the DNA sequence precisely. That's the magic of CRISPR. It's fast, it's cheap, and it's incredibly precise, at least compared to older genetic engineering tools. And this is where things start to get exciting. Let's talk about medicine first. One of the most promising areas where CRISPR is making waves is in treating inherited genetic diseases. Take sickle cell anemia, for example, a painful and often deadly condition caused by a single mutation in a person's DNA. With CRISPR, researchers can take a patient's stem cells, correct the faulty gene, and then return those corrected cells to the patient's body. In 2020, a woman named Victoria Gray became the first person in the US to receive this kind of CRISPR therapy for sickle cell disease. After the treatment, her symptoms drastically improved. That's not just a theoretical success, that's a real person whose life was changed by gene editing. And it's not just sickle cell. CRISPR is being explored for a wide range of conditions, from cystic fibrosis to muscular dystrophy to rare forms of inherited blindness. In fact, scientists have even begun testing CRISPR-based therapies that edit genes inside the human body, not just in isolated cells. One example? A clinical trial aiming to restore vision in people born with a rare genetic form of blindness. Researchers injected CRISPR directly into the eye, hoping to cut out the faulty gene in retinal cells. While the results are still early, the fact that this is even possible is astounding. Then there's cancer. One of the frontiers in oncology is immunotherapy, training the body's immune system to recognize and destroy cancer cells. CRISPR makes this even more powerful. Scientists can extract a patient's immune cells, edit them with CRISPR to enhance their cancer-fighting ability, and then reintroduce them. Think of it as reprogramming the body's internal army. Trials are already underway using CRISPR-edited T-cells to treat leukemia and other cancers, with some early success. But CRISPR's impact isn't limited to medicine. It's changing how we think about agriculture, food, and the environment. For decades, we've used genetic modification to improve crops. But those older techniques were blunt, slow, imprecise, and often involved adding foreign DNA, which made people uneasy. CRISPR changes the game. It allows scientists to make precise, targeted edits to a plant's own genome without introducing genes from other species. Want a tomato that doesn't bruise as easily? Wheat that resists fungal infections? Rice that produces more grain? CRISPR makes all of that more efficient and more acceptable to regulators and consumers. And since it's faster and more affordable, even smaller research groups in developing countries can use it. But perhaps the most surprising and arguably most urgent use of CRISPR is in the fight against climate change and ecological collapse. Some scientists are exploring how CRISPR can help save endangered species by editing out harmful mutations in small, inbred populations. Others are looking at gene drives, tools that can spread genetic changes through wild populations of pests like mosquitoes, potentially eradicating diseases like malaria. Of course, gene drives raise their own ethical and ecological questions. What happens if a change spreads in unintended ways? What if we cause harm to an ecosystem by removing a key species? These aren't easy questions, but they show just how powerful this tool really is. And yes, we can't ignore the elephant in the room: embryo editing. The case that made CRISPR infamous was in 2018, when Chinese scientist He Jiankui announced he had edited the embryos of twin girls to make them resistant to HIV. The global response was swift and harsh. The experiment was condemned by scientists, ethicists, and governments around the world. Why? Because editing the germline, the DNA of embryos, sperm, or eggs, means those changes are inherited by future generations. That opens the door to mistakes that can't be undone. It also raises deep questions about consent, inequality, and what it means to be human. Most scientists agree that we're not ready, technically or ethically, for widespread germline editing. And in many countries, it's outright illegal. But this isn't where CRISPR's real power lies. In fact, it distracts from all the safer, more impactful, and more immediate ways CRISPR is transforming the world around us. Here's the bottom line. CRISPR isn't about creating superhumans. It's about treating the incurable, feeding the hungry, and protecting our planet. It's about replacing trial and error with precision. It's about giving us tools to understand and edit life at its most fundamental level, with all the responsibility that implies. So the next time you hear someone say, "Isn't CRISPR just about making designer babies?" you can confidently say, "Not even close." That's just the headline. The real story is much bigger, and it's just getting started.